We're going to look at some past paper questions now, based on the Genetics Part 1 video. So if we look at 2019's paper, paper 2 for November, question 1.5. A certain plant species has the following alleles. Number of seeds per pod, and there you're given 2 codes. Leaf shape, and you're given 2 codes. And then the table below shows the results of the offspring, and there you can see 4 different phenotypes. Notice that each phenotype has 2 characteristics in it, but that's still considered to be one phenotype. So how many genes are being considered here? This is a dihybrid cross, and so 2 are being considered. What is the dominant phenotype? If you look at the code, 1 seed and normal shape. You've got to give both because it's a dihybrid cross. So you would write 1 seed normal shape. Give the genotype of each parent. Now again, the fact that you've got this 4 phenotypes listed and that each phenotype has different characteristics in it indicates to you that this is the F2 generation in this dihybrid cross. And if you think back to the video, the F1 generation that gives rise to the F2 generation where we have a 9:3 to 3:1 ratio in the phenotype, that F1 generation is always heterozygous. So the genotype for each parent here is going to be heterozygous. It's only if we have that heterozygous parent which was the F1 generation and then becomes the parents of the F2 generation that we can end up with those 4 different phenotypes. Finally, it asks number of offspring that are homozygous recessive. Homozygous recessive for both characteristics. In other words, if we look at the recessives, 3 seeds and wrinkled shape. Well, the only one with 3 seeds and wrinkled shape that's listed is that one, which is 32. Question 2.4. The back of the leopard frog can be spotted, as shown below, or be without spots. Spotted frogs were allowed to interbreed and they produced 150 spotted and 50 without spots. Which phenotype is dominant? Alright, so we've got spotted frogs breeding with spotted frogs because spotted frogs were allowed to interbreed. But their offspring contain both spotted and without spots. That tells you that the spotted frogs that were the parents must be heterozygous because otherwise we couldn't end up with with an unspotted frog. The fact that spotted frogs are producing unspotted frogs means that the spotted condition must be the dominant one. So which phenotype is dominant? Spotted. Explain your answer. So you would say either that because spotted frogs interbred and produced offspring without spots, or because the ratio of offspring, so you've got 150 to 50, which is 3:1. The ratio of offspring, 3:1, 75%, 25%. You couldn't get that ratio without the 75% being the dominant phenotype. So your best answer is to say you've got spotted frogs that are producing offspring that do not have spots. Right, 2.4.3. A frog that is heterozygous for spotted back was crossed with a frog without spots. Using the letters D and D represent a genetic cross to show the expected genotypes and phenotypes of the F1 generation. Alright, so we need to write out our whole monohybrid characteristics. We need to write out our whole template. So we're going to be looking at skin pattern or skin color, something like that. The key that we're going to use will be capital D for spots. Small d means no spots. And our P1 phenotype, we've been told we have a spotted individual crossed with no spotted individual, without spots. Our genotype, we were told that our spotted individual is heterozygous, and no spots is the recessive condition, which means that the genotype must be small d, small d. Don't forget to write the word meiosis before you do your gametes. which will be capital D, small d crossed with small d, small d. Notice the commas. The commas are very important. They tell your examiner that you know that these are separate gametes. If you don't put the comma in, even if you space the letters apart, your examiner or your marker is not quite sure whether you mean to write the genotype or whether you mean to separate them out as gametes. So make it very clear, just put the commas in. Then don't forget the word fertilization before you do your Punnett square. All right, so I'm going to do my Punnett square up here because I've run out of space over there. You put one parent on one side, other parent on the other side, and fill in your Punnett square. And don't forget to write F1 generation. And then we're going to give the F1, let's do genotype first. Don't know why it's gone off on that one. Right, F1 genotype, which is going to be 1 to 1, 50%, 50%, big D, small d, to little d. And your phenotype is also going to be 1 to 1 or 50%, 50%, whichever way you prefer to write it. 50% is going to have spots and 50% no spots. Now, how is this marked? These kind of questions are always worth 6 marks. There are 2 compulsory marks. The first compulsory mark is for listing F1 and P1. So that's 1 mark, compulsory. And the second is for writing the words meiosis and fertilization in the correct places. So those are 2 compulsory marks. And then there's another 6 possible marks that you could get for filling in the genotype correctly, filling in the gametes correctly, filling in your Punnett square correctly, giving the genotype correctly, or giving the phenotype correctly. 1, 2, 3, 4, 5 additional marks. Apologies. So if you fill in your Punnett square, you will get a mark. If you fill in meiosis, fertilization, P1, F1, you'll get a mark. If you then write the solution, the genotype, phenotype, you could get another mark there. It's very easy to get your full 6 marks on a monohybrid cross question. All right, let's look at another dihybrid cross question. So here we have a certain species of rabbits. The body color is controlled by 2 alleles. Capital B, black is dominant to white. Ear shape, wide is dominant to narrow. What is the possible genotype for a black rabbit with narrow ears? Okay, black rabbit is the dominant, so there must be at least 1 capital B. Narrow ears is the recessive, so that must be the 2 little e's. So now let's look at our answers. This one doesn't have a capital B, so it cannot be that answer. This one has a capital E, cannot be that. This one has a capital E, cannot be that. So therefore, it must be D. What is the possible genotype of gametes produced by a white rabbit with narrow ears? So let's write out the genes. You've got a white rabbit, white is the recessive, so that's going to be small b, and narrow ears is going to be small e. Now your gamete must have 1 of each of those. So your gamete is going to be BE, which is B. Notice it's not going to be this because that's a dihybrid cross further down. So that would be at the end of the F1 generation when you then cross-pollinate, cross-fertilize, whatever, heading into the F2 generation. It cannot be this because that is the genotype of the F1 generation. And you can't have this because there's a capital E there, which is for the dominant for wide ears. So it cannot be either of those 3, any of those 3. All right, another monohybrid cross question. So we've got flower color, purple or white, in a particular plant species controlled by the alleles capital D, small d. You've got 4 crosses and their offspring is listed there. State the dominant flower color. Okay, look at cross number 1. In cross number 1, you have purple plants crossed with white plants, but all of the offspring are purple. The fact that all the offspring are purple tells you that the moment you have one purple allele, that is the dominant, because that's what's showing up. So your dominant color here is going to be purple. Use cross 1 to explain your answer. We just did. The fact that you have purple parents and white parents, but all of the offspring are purple, indicates that purple must be the dominant. State Mendel's law of segregation. Notice that this is worth 3 marks, so you need to give a lot of detail. So Mendel's law of segregation says that when gametes are produced, the inheritable or the heritable factors of the parent are separated from one another so that one copy goes into each gamete. Thus, the offspring is able to inherit one copy of each factor from each parent. Okay, 3 marks. You need to state the whole thing. And then 244, use a genetic cross to show how the crossing of 2 purple plants can produce white offspring, such as in cross 2. Okay, so again, we're going to do the whole template. So our characteristic is flower color. Our key, D, is going to be purple. Small d is going to be white. Our P1 phenotype is going to be purple crossed with purple. And our genotype, we know that our genotype is going to have to be heterozygous if we're going to have white in the offspring. So it must be, apologies, I'm writing P's there, DD crossed with DD. Don't forget to write the word meiosis before you do your gametes. So the gametes for each are going to be big small crossed with big small. And then again, write the word fertilization before you do your Punnett square. Big D, small d, big D, small d. Don't forget to write F1 generation. So then we've got our F1 genotype is going to be 1 to 2 to 1 because I've got 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. The phenotype, however, is going to be 3 to 1, or 75%, 25%. So 3 to 1, purple to white. And again, your marks allocated for this are going to be, for your P1 and F1 together, is 1 mark. And for meiosis and fertilization together is 1 mark. Those are compulsory marks. And then, depending on where else you've allocated, additional marks there, making up to 6. Right. This is in section A, and we've got 3 questions here based on this diagram. So here we have one pair of homologous chromosomes. On 4 fruit flies, the alleles for antenna shape and body pattern are indicated. So you've got a nice key there. Which fly is homozygous for antenna shape and heterozygous for body pattern? So if you have a look at the antennae shape, homozygous means they must be the same. So we're looking for ones which are the same. So either this one or this one. Which of those 2 are heterozygous for body pattern? So your body pattern at the bottom lots, they must be different. This is the one that is different, and so therefore fly number 2 is the correct answer. What is the genotype of fly 3? Okay, so for fly 3, all 4 of these patterns, if you look at them, are different. So it's heterozygous for both conditions. So if we write out the heterozygous for both conditions, we will get this. So let's find that in the answer. And it is this one. Heterozygous for both conditions. Which one of the following will represent the possible gametes for fly number 4? All right, if we look at fly number 4, we've got a heterozygous condition for the antennae and a homozygous condition, and it's recessive, for the stripes. Okay, so let's write that out. So you've got heterozygous, homozygous. Big N, small s, small s. So now let's go down and we're looking, so we've got, let's just write that here again. So we've got it, big N, small s, small s. One of those ends, remember first outer inner last, one of those ends must go into each gamete and one of those s's into each gamete. So our gametes are going to be big N, small s, or big N, small s, or little n, little s, or little n, little s. Okay, first, outer, inner, last. We do away with the repetition, which leaves us with big N, small s, small n, small s, which is D. Right, let's have a look at this next question. Section A question, so the answers are all pretty short. Mendel observed some characteristics of the pea plant, which he suggested were controlled by inherited factors. He conducted a series of experiments in which he crossed pea plants with contrasting phenotypes to obtain the offspring of the F1 generation. At first, his crosses were simple and involved only one pair of characteristics. Mendel counted the number of offspring showing each of the variations, and his results are shown in the table below. So we'll go through that table in a moment. Give the term for the inherited factors that Mendel referred to. So your inherited factors are your genes. A cross involving only one characteristic is known as a monohybrid cross. The female structure of the flower where meiosis occurs is the ovary. Use the information in the table above to give the number of each of the following. The characteristics of the pods. So if we have a look at the pods, you can see that there are 2 different characteristics. So we have these 2 characteristics. The alleles for seed characteristics. So if you have a look here, under the seed characteristics, the P1 generation, that gives you the alleles. So you can see we've got 1, 2, 3, 4. Give the characteristic that is dominant for flower color. So if we scroll up and we look at the flower color, you can see you've got a violet flower crossed with white, and the offspring are all violet, which means that violet is the dominant color, because white will only show up if there is no dominant present. And then they ask for the recessive for stem height. So if we look at the height of the stem, you've got tall crossed with short, and all the offspring are tall, which means that short must be the recessive characteristic. We don't see any that have the short characteristic, which means that that must be the recessive, only visible when there is no dominant present. 145 says, if the individuals of the F1 generation are crossed, how many phenotypes for seed color would be expected in the F2 generation? So if we look at our flowers, so if we look at our seed color, so we're looking at this one, you've got yellow crossed with green, and all the offspring are yellow in the F1 generation. That means that they must be heterozygous. So we're taking 2 heterozygous parents and crossing them to get our F2 generation. So we've got, let's say we're going to use Y. So we've got big Y crossed with little y, big Y crossed with little y. Okay, how many phenotypes have we got there? Well, if we look at the genotypes, we've got a 1 to 2 to 1 for the genotypes, because we've got 1 big Y, 1 big Y, small y, sorry, 2 big Y, small y, and 1 small y, small y. But they're asking about phenotypes, so I'm looking for the dominants. There is a dominant, and there is a dominant, which means I have 3 to 1, and those will be yellow to green. Okay, now that still hasn't answered the question, because the question asks how many phenotypes would be expected, and I've got 2 phenotypes here, so the answer for that is 2. Right, let's have another dihybrid cross example. So, from November 2016, the leaf color in a plant is controlled by 2 alleles, green and yellow. Thorns are controlled by 2 alleles, present or absent. 2 plants with the genotypes capital G, capital G, capital T, capital T, and small g, small g, small t, small t were crossed. Their offspring were then left to pollinate with each other. Okay, so if they're crossed, that's your F1 generation, which then are left to pollinate each other. In other words, that's your F2 generation. The table below shows the possible genotypes of the offspring of the second generation. Genotypes 1 and 2 have been left out. So there's genotype 1, there's genotype 2. Give the genotype of 1. So this is very straightforward. We're just going to use our Punnett square. We're going to put these 2 gametes together. Remember that we always put the capital letter first. And so it's going to be big G, big T, small t. And give the phenotype of 2. Well, obviously, we've first got to work out what the genotype is. So the genotype for this one is that gamete plus that gamete, which would give us small g, small t, small t. Now we need to translate that into the phenotype. Well, small g's are yellow, and the small t's are no thorns. Question 2.1.2. List the 4 genotypes of the offspring of the second generation that would be phenotypically different from the original pair of parents. So we do not want this, and we do not want this. Okay? What we're looking for, so the phenotypes for these 2, for this parent here, is going to be green. Let me just swap pens. Is going to be green with thorns. This one is going to be yellow with no thorns. So what I'm looking for now are genotypes that are going to give me a different phenotype. Because you only need 1 dominant letter present in order for the dominant characteristic to show, essentially what I'm looking for are genotypes where there are capital letters in one of the 2 conditions, but not in both. So for example, in that one, I've got a capital G, small g, so it'll be green, but it has small t, so it'll have no thorns. And that is different phenotypically to both parents. Likewise, that one, that is going to be yellow with thorns. This one is also going to be yellow with thorns. This one is going to be green with no thorns. And this one is the same as the previous one. And this one is going to be green with no thorns. All the others have capital letters in both the color and in the presence or absence of thorns. So now, when we look at our answer, it says list the 4 genotypes. So we're going to have capital G, small t, capital G, small t, small t, small g, capital T, and small g, capital T, small t.